You may note that my research group has had a long history of interest in delivery of modulators of drug resistance in cancer, and that is because we think targeted delivery of these agents to cancer site is very important, because we don't want normal tissues and cells get more sensitive to chemo or radiation therapy. So recently we have actually been focusing on another mechanism of drug resistance, and that is DNA repair. So that brings it to my talk on nano-delivery of novel inhibitors of DNA repair for targeted chemo and radio-sensitization of solid tumors. This research has been actually targeting an enzyme that is involved in DNA repair and has been recognized as a novel target for inhibition of DNA repair in cancer cells. The research on this target, which we abbreviated as PNKP, standing for polynucleotide kinase phosphatase, started in the laboratory of Dr. Michael Weinfeld at the University of Alberta a long time ago. Michael has been working on validating this enzyme, this DNA repair enzyme, as a drug target for a long time. Later, he joined forces with Dr. Dennis Hall from Department of Chemistry at the University of Alberta to develop a small molecule inhibitors of this enzyme as new therapeutic agents for cancer treatment. Let's see what PNKP does. If the DNA gets damaged through exposure to ionizing radiation, reactive oxygen species, or topoisomerase 1 inhibitors, we can end up with DNA strands that have flipped parts. What it means that their 3' end is phosphorylated and their 5' end is dephosphorylated. PNKP is an enzyme that has both kinase and phosphatase activities, so it can basically repair this kind of defect. And then when this defect is repaired, the DNA strands can be rejoined through the action of DNA polymerase and DNA ligase. Now, if PNKP is inhibited, it is rational to think that the cancer cells may become sensitive to the effect of the DNA damaging effect of ionizing radiation and topoisomerase 1 inhibitors. So, as I mentioned, several different inhibitors of PNKP have been developed by Dr. Dennis Hall in collaboration with Dr. Michael Weinfeld. And from these several agents, we have chosen one that had relatively potent characteristics in the inhibition of PNKP. So this hit molecule was abbreviated as A83B4C63, and from now on, I'm going to refer that as A83. So we made a hypothesis that nano-encapsulation of this molecule in colloidal systems or nano-delivery systems would be able to increase the water solubility of this drug or this drug candidate so that its IV injection would be possible in animal models, because this compound was very water insoluble. And our second hypothesis was that nano-delivery of A83 by appropriate nano-delivery systems can make them more potent and also more specific in sensitization of solid tumors to the effects of ionizing radiation and also topoisomerase 1 inhibitors. For the delivery of this compound, we chose polymeric micelles, and the reason was, again, because of the poor solubility of this compound and hydrophobic nature of this compound. So we thought that polymeric micelles are better basically delivery vehicles. So as building block of polymeric micelles, we chose to work with a polymer platform that has been developed in my lab some time ago by a former student in my lab, Dr. Abdullah Mahmoud. As I mentioned, the original paper on this platform has been published a long time ago in 2006. So this platform is based on polyethylene oxide and polyesters, particularly polycaprolactone, that are functionalized on the alpha carbon with different groups. This polymer platform has several advantages. First of all, we have the polyester backbone that is biodegradable and has a history of safe application in human. We can lyophilize this polymeric micelles, unlike the original polycaprolactone, and that is because the introduction of this functional group has basically made the polymer amorphous, so that then it can be lyophilized by freeze drying and then reconstituted. We can introduce chemical flexibility to the polyester structure just by substitution of different R groups on the polycaprolactone, and that can make this system amendable for encapsulation of different chemical entities. Over the years, through the work of two postdoc fellows in my lab, Dr. Xiaobing Xiang and Dr. Mohammad Reza Wakili, we have generated a library of these polymers having different R groups in the core and also different functionalities and ligands on the shell of the nano-delivery systems. This polymer platform, which was developed at the U of A, has been licensed to Meros Polymers, a spin-off company that I am involved with. For the sake of this project, we chose to use the polymer with the benzyl carboxylate in the core because of our previous experience with this polymer and its excellent properties in solubilization of hydrophobic molecules. So we did encapsulate A83 using this benzyl carboxylate substituted polycaprolactone block copolymer that I am going to call it POPBCL from now on. So this is basically the TA pictures and also DLS data. Similar to several other hydrophobic molecules, what we noticed was that the release of A83 from the POPBCL was reduced drastically, and this was in contrast to the formulation of this drug in the POPCL micelles. We then looked at the in vitro activity of A83 as encapsulated drug compared to the free drug in sensitization of colorectal cancer cells to ionizing radiation. So this graph, you see that this is colony forming assay, and I wanted you to notice that this is on log scale. So the black line actually shows empty micelles just with radiation without any A83. And then the green and red one shows addition of A83. So you see that it was able to sensitize cells to the effect of radiation. And similar data was found for irinotecan, which is a topoisomerase 1 inhibitor. Later, we used sRNA against PNKP to compare the results we got with A83 with downregulation of PNKP, and we found that the results were pretty similar. So our drug was able to sensitize the cells to the effect of ionizing radiation and irinotecan, similar to the sRNA against PNKP. Then we went in vivo, and the first thing we did, we tried to come up with a formulation using conventional surfactants that can solubilize this drug for in vivo administration. And we ended up using creme4EL and ethanol formulation. And then we looked at the safety of this compound in healthy mice and identify the maximum tolerable dose of this compound in healthy mice. This work that has been done by Sam Sadat, a graduate student in my lab who recently defended his PhD thesis, is part of this paper that has been recently published. What we found was that the drug, as the Chemo-4EL formulation or nanoparticle formulation, was safe and did not cause any weight change or any biochemical changes in different factors or parameters that we have looked into, up to 50 milligram per kg. And that was the highest dose that we tried. In the next step, then we look at the effect of this drug and its nanoparticle formulation in radio-sensitization of cancer cells. Again, we did some in vitro work to assure and make sure of the mechanism of action. So in this slide, what you see is the analysis of gamma H2AX staining, again, in colorectal cancer cells that were exposed to radiation only, and a combination of A83 in nanoparticle form and Chemo-4EL formulation with the radiation. And as you can see, A83 definitely shows an increase in the staining of gamma H2X, which means that we have actually more DNA damage or more DNA double strand breaks in the cells that were exposed to combination therapy. The next step was to bring this formulation to animal models. So in this study, we used HCT116 mice, a subcutaneous model, and then exposed the tumor to radiation. But before exposing the tumor to radiation, we injected the drug either as Chemo-4EL formulation or nanoparticle formulation 24 hours before the radiation. And this was basically the schedule and the design of the study. Our results actually showed that among different groups that we had, only the nanoparticles of A83 were able to sensitize the tumor xenografts to the effect of ionizing radiation. And the free drug, or the drug that was solubilized with the aid of Chemo-4EL, was not able to do that. We confirmed these results through measurement of tumor volume by caliper measurement, looking at tumor weights, and also measuring the uptake of FLT as a biomarker of tumor cell proliferation, or a PET contrast agent showing the proliferation of tumor cells in vivo. And this also basically confirmed our caliper measurement data, as the nanoparticles of A83 were the only ones that have shown a lower proliferation of the tumor cells by FLT imaging. We also looked at the IVIS imaging of the tumor and made a comparison between tumor luminescence before treatment and after treatment. And as you can see again, this confirmed our previous data. The only group that did not show a substantial growth in the tumor was the one that was injected with the nanoparticles of A83 in combination with radiation, and free drug, or the drug solubilized with the aid of Chemo-4EL, was not able to do so, and that confirmed our previous data. So then we basically studied tried to find out what is the reason for this. So we looked at the biodistribution of the drug following injection of Chemo-4EL and nanoparticle formulation. And what we found was very interesting. Well, our findings showed this is the plasma profile of nanoparticle versus Chemo-4EL, and this is the tumor profile. So we found out that actually at 48 hours, we see a kind of delayed accumulation of the nanoparticles of A83 in the tumor. And at this time point, basically the drug is, cleared from all other major organs except from the plasma and the liver. So that is a surprise, but it's a pleasant surprise because that enables us to basically time the radiation perfectly or the chemotherapy perfectly, where we don't have accumulation of the drug in healthy tissues, but we have high concentration of the drug in the tumor tissue. So after this, now the second part of this project was to look at the effect of combination with chemotherapy. I have shown you some results, in vitro results before, but in the continuation of the project, instead of looking at irinotecan, we tried to look at SN38, which is the active metabolite of irinotecan and is 1,000 times more potent than irinotecan. In this project, we actually developed a nano-delivery system for SN38 first by conjugation of SN38 to the end of POP-BCL, so chemical conjugation of SN38. And then we used this delivery system either as a separate entity to combine with A83 nanoparticles or as a nanoparticle that can co-encapsulate A83 in the core as well. We did the characterization of these systems. So the ones that are highlighted here are delivery system for individual drug, SN38 or A83, and the one in the middle is the nanoparticle that co-encapsulates both drug in one nanoparticle. We have done some preliminary cytotoxicity study on different colorectal cancer cells, but I just selected a few of these graphs to show you here to convey the message better. So as you can see, this graph shows the two separate SN38 and A83 nanoparticles that are combined together. So in this case, we see that both as separate nanoparticles or free SN38 with nanoparticles of A83, they were able to make the cells sensitive to the effect of either free SN38 or nanoparticles of SN38. And this was very pleasant. We also compared the co-delivery system, and the co-delivery system was also able to make the cells sensitive to effect of SN38. But at this point, we cannot really make a direct comparison between the co-delivery system and the combination of the two individual delivery systems, since the concentration of A83 was different in the co-delivery and the combination. And we have to do further in vitro, and I think most importantly, in vivo studies to find out which one provides a better opportunity for synergism between these two drugs. I also wanted to mention that recently we have actually tested this combination in other cancer cell lines, including head and neck cancer and lung cancer cells, and we have found similar results in different cancers. So we can extend our investigations to other solid tumors. At the conclusion, I wanted to say that we found that the free and polymeric micelles of A83, they acted as chemo and radiosensitizers in several solid tumor cell lines in vitro, but in vivo, it was only the polymeric micellar formulation of A83 that was efficient and was working, and the free drug solubilized with the aid of Chemo-4EL did not work. I hope that this data was able to convince you that in addition for drugs that are in use, nanomedicine may be used to basically enhance the profile of drugs that are in development. As we have seen here, if we were going with the Chemo-4EL formulation for A83, we would have thrown this drug candidate away. but with nanomedicine, it worked perfectly. I wanted to thank people in my lab, and the ones that are highlighted here are the ones who have contributed to the work presented in this talk. Also wanted to show you some pictures from our lab, and this one was taken a few days ago. And also thank the funding agencies and the collaborators. Again, the ones that are highlighted here have contributed to this work. And I'm ready to answer any questions.
Okay, thank you, Afsaneh. So maybe quick answer to this question. Have you seen any influence of these nanoparticles on secondary tumors?
Yeah, well, we have actually done some studies on orthotopic model of colorectal cancer where the tumor has metastasized to other tissues, if that's what is meant by secondary tumors. And we have seen some effect in that. Also, we have some recent evidence that this combination of radiation and PNKP inhibitors may activate basically immune cells against or make the tumor cells immunogenic. So there is also a potential for that, but that needs more investigation.
Okay, thank you very much, Afsaneh. So if there are other questions, maybe you can answer online.